Well, thank you so much for being here in this spotlight interview with Dr. Shireen Mentor. She holds a PhD from the University of Western Cape, and she is currently based at the University of Cape Town in South Africa, in the capacity of both academic lecturer and researcher. Her current work focuses on vascular endothelial research to elucidate the underpinning mechanisms of blood-brain barrier disruption in the context of disease. She is trained in cellular biology, molecular biology, and electron microscopy, with a particular interest in the underpinning physiological and pathological mechanisms that facilitate or alter the process of endothelial barrier establishment. Shireen is a recipient of both national and international awards, one of which is being a Fulbright alumna of the University of Missouri as a visiting research scholar in the Department of Neurology, Center for Translational Neuroscience, where she was afforded the opportunity to interrogate the blood-brain barrier by utilizing cutting-edge imaging tools, which enabled her to, for the first time, document the snapshot development of the blood-brain barrier and extrapolate ways to illuminate future treatment intervention for neurodegenerative diseases. Today, we will be discussing her PLOS ONE Lab Protocol article entitled Preparation of Biological Monolayers for Producing High-Resolution Scanning Electron Micrographs, and the associated protocol in protocols.io. Shireen, thank you so much for talking to us.
Thank you so much, Gabriel, for having me.
To begin, I would like you to tell us briefly what this protocol is about, how you use it, and why it is relevant.
Okay, so essentially the protocol is a comparative study where we looked at a metal alloy coating versus a non-metal coating. We looked at how those coating methods, what kind of morphological data can come from those coating methods when looking at specifically brain endothelial cell development. And so the reason why we find this to be relevant is because the complexity of the topographical structures on an endothelial cell may likely play a part in its ability to reinforce barrier properties, okay? So the brain endothelial cell is essentially the anatomical basis of your blood-brain barrier. And your blood-brain barrier's role is to control and regulate the ionic concentration in your brain's microenvironment, maintain homeostasis, and also it's really important for our psychological aptitude. And so for us, it was important to understand how does this endothelial cell become so complex? And why is it so robust? And then morphologically, what can the morphological data tell us about how it endorses that restrictive barrier properties? And so that was really like the motivation and the relevance for the study, but also in choosing the correct metal alloy or coating for studying these morphological structures at high resolution is very important, because if you choose the wrong coating, you might lose all of the salient features that are actually necessary for barrier interaction in endothelial cells.
I see. And can you then describe what was the state of the art at the moment when you decided to create and optimize this protocol? In other words, was there anything similar already published? And if so, why those protocols didn't work for you?
Okay, so yes, there were similar coating methods published, but we can't just choose an arbitrary coating method and apply it to just any biological sample, right? You have to understand what you're looking for. And so a lot of the previous protocols had chosen coating methods, for example, that were just gold coating, metal coating, and then there were non-metal coating methods such as carbon, okay? And what we found was that in looking at some of these carbon micrographs, we found a lot of 2D structures, okay, with a lot of these ultrastructural nano features missing. And so this 2D structure makes a carbon coat seem more appropriate for dimensional investigations of a cell, right? And so this was really the motivation for us to maybe look at a gold palladium and then try and optimize that gold palladium coating in order to enhance three-dimensionality or sort of help us profile that detailed structures more clearly.
And after all the time that it took you to develop experimentally this protocol, what was your motivation to make it public as a protocol in protocols.io?
Okay, so essentially for us, it was just based on the fact that we'd seen all of these structures that had these protocols that had shortcomings when it came to coating biological samples. We thought, okay, you know, if we make our protocol more accessible to some microscopists who are studying endothelial barriers or monolayers, cellular monolayers, this might be beneficial to them because they might be missing a vista of features that they are unaware of. You know, and so when we discovered these structures, to us it was uncharted territory. It was novel. You know, for the first time we saw nanovesicles, we saw nanotubules, we saw cells hemifusing with each other, which you don't really see in a carbon coat. And so we thought, let's choose PLOS ONE because PLOS ONE is essentially it's highly utilized, and so it really gives us it's an ideal platform for us to reach more scientists studying using high resolution electron microscopy.
And can you describe if and how the feedback that you received from the peer reviewers at PLOS ONE shaped the final version of your protocol?
Essentially, the peer review process was constructive criticism, and we opted for that peer review process because we needed to understand where we were being redundant in the protocol and if the work was actually going to make an impact, if there was any sort of novelty that we could add to this existing body of work in terms of preparing biological samples for high resolution microscopy. And so that sort of feedback, you know, that came, that fine-tuned our protocol, was quite beneficial. We found that to be, you know, a quite beneficial process, I would say.
And I would like us now to delve a little deeper in the protocol itself. And for the users, is there anything that you tried while developing the protocol that didn't work and didn't make it to the final version of the protocol? Are there negative results?
No. So essentially, I started off working with the gold palladium, right, which was at a 60-40 ratio, and we saw a lot of detail, and we thought that we could maybe just fine-tune that particular method to see if we could really extrapolate more high-resolution nanoscaled features. And then we ended up choosing a carbon coat, okay? And that was when we actually saw, oh, there's a big discrepancy between the two types of coating. And then we went back to gold palladium and we went into an 80-20 ratio. And so there was this three sort of coating methods we were sort of using to find the best three-dimensionality within our particular brain endothelial cell. And so, yeah, that's essentially what we did in terms of optimizing. We wanted to find as much of these nanostructural features as possible and sort of galvanize a step-by-step snapshot development of your blood-brain barrier, essentially.
If the user wants to start using your protocol, what are the most critical steps for a successful experiment? The most critical steps are making sure that your buffers and your fixative solutions are actually within the correct osmolality range, okay? So when you're using any particular cell, right, you have to understand what environment that cell comes from in the body, okay? So now if we're dealing with your brain endothelial cell, which is the basis of your brain capillaries, it's exposed to your blood plasma. And so we had to create buffers within an osmolality range of about 275 to 290 milliosmoles per kilogram, okay? And this helps us create an isotonic solution. Because if the osmolality of those buffers are completely incorrect, right, what you're going to have is you're going to have a
hypertonic solution, okay, where you're going to have perhaps fluid leaving the cell and you're having crenation of your cell sample, right? Or you might have a hypotonic solution where you have water entering the cell and the cell swells. So now we want to preserve the cell in its most native state. So osmolality is actually very important, right? And also the time of fixation. If you want to fix your sample, your monolayer, ensure that it is fixed at room temperature for 2 hours, maximum 72 hours, and in a refrigerator. If you go over that time, you'll find that the sample is somehow distorted.
I see. So, and
Yeah.
those are the two most critical steps: the solution
Yes.
and the fixation.
Yes, those are very important steps
And
to ensure a native state.
Since you published the protocol, has there been any further optimization or any changes that you have implemented to the protocol?
No, there has not. One thing that I do believe as a researcher, as an emerging researcher, is that once you've developed a really good method, it forms a really good foundation for the rest of your study, you know? And so we didn't need to develop anything else. And the beauty of this particular protocol is that I've applied it to different chapters of my PhD dissertation, and we published 3 more papers, morphological papers based on this protocol. So no changes.
That's really amazing and really good
Yeah.
to hear. So actually, I wanted now to tell us what has been the significance of developing the protocol and publishing the paper in your young career as a scientist. And you already, sort of like answered the question, but has publishing this paper had any other positive impact
Yes, it has.
in your career?
It has had tremendous impact, actually. So the protocol actually became the basis of my PhD study, right? And then, as I said, 3 other publications, morphological related publications were then published through using this particular method. This PhD was then published as a dissertation by publication, and then won best PhD thesis by the MDPI Healthcare Journal.
Oh, congratulations.
And another additional award was the Council of Laureates at the Nobel laureate meetings then recognized the novelty of these high resolution images, and I was fortunate enough to present the work at the Lindau Nobel laureate meetings in 2018.
Oh, congratulations. I'm
Thank
really
you.
happy to hear that.
Thank you.
Yeah, no, really, really impressive. And so proud of very tangentially be part of that process.
Thank you so much.
But and before we wrap up, is there anything else that you would like to mention that we haven't touched so far in this conversation?
I think for me, what I'd like to maybe mention is that, you know, PLOS ONE has actually set up the electron microscopy units at UWC, University of the Western Cape, South Africa, and the neurobiology research group at the University of the Western Cape in such a way that our work is now no longer siloed to only our labs, but it's now got an international audience. And so in that sense, you know, you've sort of positioned us in a very, very favorable way to perhaps collaborate or find collaborators through this platform. And so for that, I'm quite grateful.
Thank you again, Shireen, so much for sharing your time and your knowledge and your insights with us. And I bet it's going to be very useful for the users of the protocol.
Thank you so much, Gabriel.